Hi everybody, thanks for joining us for this presentation. I'm going to talk about cross-contamination and tips to avoid it in the histology lab. So we're just going to go station to station, and we are going to talk directly about those stations and small little things that we can do to avoid cross-contamination. So first of all, at the growth station, there's a couple of things that you can do as you're grossing along in order to prevent specimen-to-specimen contamination. I always keep just a little squirty bottle of 70% alcohol at my growth station, and I wipe down my growth board with the 70% alcohol. It's just really simple. I just use a paper towel, wipe it down, and then go to my next case. I also will keep a 70% alcohol container, whether that's a little cup or a little flask of alcohol, right at my growth station. And my utensils that I'm using, I'll just drop in there when they're not in use, and that gets all those little free-floating particles from previously cut cases coming off of your scalpel or coming off of your scissors that you're using. And always, as a good rule of thumb, only open one specimen container at a time. Oftentimes we'll have maybe a growth assist that's with us, and it's just a really good practice to get into at the growth station on the table. We have one container open. We put that specimen back in that container when we're done, close it up, and then go to the next one, as opposed to having 4 or 5 different containers lined up and open, ready for that growth tech to do their job. Another thing to consider at the growth station is when we're running the bowel. Running the bowel is a term that's used when you just take your scissors and run down the tube of the bowel and expose that mucosa. Fecal material is oftentimes in that mucosa, so a nice good washing of that mucosa can be done. If you have any remaining fecal material that's left, you can possibly get vegetable or cellulose matter in that section. If you do have any kind of vegetation or cellulose that's left in the mucosa of the bowel, this is an example of what that would look like. And if you are, right now what you're looking at is a PAS with a light green counter. And if you look at this with an H&E, that cellular material can show up. But if you're not 100% sure that it's actually cellulose, you can run a PAS on it, and that vegetation cellulose will come up with a positive PAS reaction. Then we go to processing. Processing, we should all be in the habit of wiping out our retort of our processors in between runs, and of course checking your solutions often if you're running more than one run a day, or daily if you're just running one overnight. We are looking for, with our solutions, the precipitation that can occur, and your solutions will become discolored with blood products. And you can also, if you're starting out with a less than 70% alcohol on your processor, you can develop a white precipitate that will happen in that first alcohol. And we need to get that out of our retort and out of our solutions, because it can actually wind up in your specimen and then create this sort of scraping effect as you're cutting that particular specimen. Of course, we always want to run a clean cycle after each run, and by doing this, we are going to prevent those little floating particles from the solutions from entering into other cassettes. Then we go to embedding. A couple of little tricks that I've learned over the years about embedding is wiping my forceps between cases. Usually your embedding center is going to have 2 or 3 of the little forceps wells on it. And if you are opening a cassette and you're touching that cassette and that tissue with your forceps and then opening the next one, you're taking that ability to transfer small little specimen particles from one tissue type to the next tissue type. So if you get in the habit of wiping your forceps in between cases, you're going to prevent that spread from one to the other. A trick that I've learned over the years is those old towels that we have hanging around our house that have strings on them that we don't know what to do with. I just bring them into the lab, and I will lay a towel over my lap when I'm embedding. And when I'm done with the case, I'll just wipe my forceps on my towel and then stick it back into the forceps well holder. Those well holders for your forceps, however, after time, they are going to get all kinds of little tiny particles, little tiny tissue samples inside of them. So what I do is I keep a little cup full of Q-tips at my embedding center, and I will just periodically throughout the day, if I've got some, 200-300 cases that I'm embedding, I'll just pick up a Q-tip and wipe out the forceps holders, and you would be amazed at how much stuff accumulates in the bottom of those that you can transfer from your forceps into different specimens as you're going along if you're not cleaning that holder. Then we have our hot plate. If you just get in the habit of, I tear up little pieces of paper towels and have them next to my embedding center, and if I've embedded a great big 25-block case, and then in between cases, when I'm done with that, I'll just wipe off that hot plate and move on to the next case. And just like with growth, only open one cassette at a time. If you're laying 2 or 3 cassettes out on your hot plate, and even if they are the same case and you're opening them all up at the same time, you're still taking the risk of putting the wrong tissue in with the wrong case. So by wiping our utensils and keeping the holders clean, we'll prevent small particles of unwanted tissue from being placed into another block, and only opening one cassette at a time will prevent specimens from being placed in the wrong cassette. Then at cutting and microtomy, one of the things that, just in my teaching years, one of the things that I have learned is that it's very simple to forget about your utensils. We all know that we need to clean our water baths, but our utensils, our little paintbrushes and forceps that we use, we tend to just throw right back into the bucket that we, hold them in and forget about them. So I train my techs to, when they clean their water bath, to also clean their utensils. You can, pick up small little particles on your paintbrushes and on your forceps that you're using to cut, and those are going to carry over into other cases. I also train my techs to wear gloves when they're cutting. This is going to prevent epithelials from your fingers getting all over your slides. That's especially important when we start talking about immunohistochemistries with things like keratins and stuff, because those can turn out to be positive. So we want to prevent those epithelials from getting onto our slides. I also run across the question, well, I don't understand how I'm getting epithelials all over my slides when I'm wearing gloves as I'm cutting. A couple of things there. One, if you're wearing gloves and let's say you wipe your face or you scratch your head, you have just put epithelials on the outside of your gloves. So you got to be somewhat diligent about what it is that you're touching when you're wearing gloves when you're cutting. And the other thing is that there's all kinds of stuff floating in the air. So if you're not using a box of slides, Yeah, get in the habit of just putting the lid back on the box of slides so that you're not getting particles, dust particles and things dropping onto your batch of slides. Yeah, another good thing to get in the habit of is to only touch the coated end of the slide. Human finger oil as well as EPIs can be transferred onto the slides. As far as our water bath goes, we need to clean our water bath daily and bleach it monthly or weekly. I tend to do it weekly just because that's my nature. But if you are just cleaning your water bath daily with soapy hot water, you might get yourself in the position where you got things floating around in there. So if you're going to bleach your water baths, you can just get yourself a tub and make a 10% bleach in your tub, and then if you have one of the water baths that have the Corningware in them, just drop the whole thing in 10% bleach and then rinse it with distilled water and let it air dry. I also always train my techs to, if they have bleached their water bath, when they put it back onto the heating element, to flip it upside down. That way the next person that comes behind them will know that that was bleached the night before or by the person who was just at that microtome. Of course, we all know to wipe the debris from the water bath with a chem wipe between each case, and only place one ribbon at a time on your water bath. Now that can get a little bit sticky if we're talking about cutting, say, 3 levels on a block. Then of course you could potentially have 3 ribbons on your water bath while you're choosing your best sections. But my rule of thumb is 1 block, 1 ribbon on the water bath. However many ribbons you need on the water bath, as long as it's from the block that's in the microtome. Another thing to keep in mind is to clean the faceplate of your microtome. I just go down to my local hardware store and I'll purchase those disposable cheapy paintbrushes and have one at each microtome, and periodically I'll just pick up that bigger paintbrush and brush off all of the debris off of my faceplate so I'm not picking it up as my ribbon is coming off of my knife. EPIs, back to EPIs, keep your fingers out of the water bath. If you're not cutting with gloves on and you're constantly sticking your fingers in the water bath, then you're going to get free-floating EPIs in your water bath and you're going to pick them up on your slide. I have a habit also of when I pick up my slide and I put it in my water bath to go underneath of my section, I'll actually dip it up and down a couple of times just to kind of wash my slide a little bit. And that takes off that dust particles that are floating around in the air before I pick my section up on my slide. And if you have a tissue explosion on your water bath, change your water out. We need to prevent those floaters from getting onto the next specimen that we're going to cut. So I see the big breast specimens that come through and they explode on the water bath, and we pick them up as quickly as we can and get the best section that we possibly can, but then we tend to go to the next case and not think about what could potentially still be left in our water bath. This is an example of red blood cell explosion. Red blood cell explosion can occur when your water bath is way too hot. It's just like the high fat content type situation where it's going to blow up on your water bath. Red blood cells can get away from you too. Most of the time when you are cutting and your water bath temperature will spike, it's because you've got water underneath of your Corningware and in between your heating element and your Corningware. And if that's the case, then your water bath is going to spike high and then drop back down low, and spike high and then drop back down low. So you can potentially have 4 or 5 specimens that are going to cut beautifully, and then all of a sudden you've got explosion, and then, oh look, these guys cut beautifully. It's because your water bath is spiking. So we need to make sure that we are checking our water bath temperatures regularly, not just once a day, but throughout the day as we're cutting. Slide labeling. I recommend that everybody use xylene-resistant pens, but I have learned over the years that with each box, you need to test a pen that comes out of that box. Every time I open a new box of xylene-resistant pens, I'll just put an X on a slide. I'll dip it into some alcohol, dip it into some xylene, and make sure that it's still there. I have ran into the experience before where that hasn't been done, and we stick a batch of slides onto the stainer, and lo and behold, all the labels disappear. And then you got to go back and you got to check your blocks against your slides and re-verify everything. So just test your xylene-resistant pens before you use them. If you're using lead pencil, you need to be aware that lead can wash onto your slide. Not to mention that the lead pencil, the more you touch the tip of that slide, it smudges and you may have a label that's difficult to read. So always check, and recheck your blocks against your slides, and be aware that lead pencil can provide you with a washing onto your slide. Here we have pictures of, on the left-hand side, you can see a couple of eosinophilic epithelials that are sitting on top of a specimen. And again, that comes from fingers, touching fingers all over the slides, or fingers in the water bath, or fingers all over your slides, dust particles in the air, not covering your slides. And then on the right-hand side, we have an example of what human finger oil looks like. Here are examples of dust particles. On the left-hand side is a dusty slide, and you can see the long kind of stringy dust particles there. It's easy to tell the difference between dust particles and pencil lead if you just take a lens paper and wipe off the top of your cover slip. If it disappears, guess what? It is dust particle. If it doesn't, then it's under your cover slip and you might be looking at pencil lead. On the right-hand side, we have an example of what pencil lead looks like when it washes off and contaminates your slide. And if you're unfamiliar about what pencil lead can look like, really easy test to do. You just take a slide, take a pencil, write on your slide, and then look at it microscopically, and it appears as these small little smudgy black particles all over your slide. Staining contamination. Basically we're talking about H&E staining here. So when it comes to H&E staining, we need to change and rotate our solutions daily. And filter your eosins and hematoxylins daily. Hematoxylin, as it's exposed to air and sunlight, will oxidize into hematin, and it appears as a metallic sheen on the top of your hematoxylin. Once that metallic sheen occurs, then you can get this artifact on your H&E that will appear as little black, dark purple speckling all over your H&E. Filtering your solutions is also going to prevent debris from remaining on your slides that may be floating in your solutions. Keep an eye on your xylenes. Your xylenes, and we're talking about clearing and deparaffinization. If your deparaffinization xylene is starting to turn a yellowish color, then your xylene is becoming contaminated or saturated with paraffin. Yeah, once it becomes saturated with paraffin, then you can experience incomplete deparaffinization, and you're going to wind up with a blotchy stain. Yeah. As far as your clearing xylenes go, if you're checking your clearing xylenes throughout the day and you see this bubbling effect on the bottom of your xylenes, then you have water carryover from your alcohols. Basically, your 100% alcohols in your H&E line are no longer 100% because you're dragging water through from your 95. So keeping an eye on that water content on the bottom of your xylene. If you experience it, you need to change your 100%. If you're consistently experiencing this over and over and over with each run, then you need to revisit your actual H&E protocol. It could be that you're not spending enough time in your alcohols in order to remove all of that water from your specimen before you're hitting xylene. This is an example of water contamination from carryover into our xylenes. On the left-hand side, it's a close-up and high magnification of water that's remaining on the slide. And then on the right-hand side, as you see the mucosal layer of the bowel, and you can see all the little black speckling on the lower right of that picture. That is a lower magnification of water carryover. Hand cover slipping. Okay, so not many of us still do hand cover slipping out there. I'm sure most of us have the either tape or glass cover slippers by now. But sometimes there are situations where you do need to hand cover slip. Frozen sections might be a example of that. When you are hand cover slipping, and you hold your slide at a 90-degree angle against a paper towel, always chase your bubbles down towards the paper towel instead of up. If you chase them up and there's nothing there to absorb the mounting media that you're squishing out from underneath your cover slip, then that mounting media is going to come up and come over the sides, and you're going to have excess media all over your slide. If you have a stringing effect to your media, it's because the bottle basically has been left open. Xylene or toluene-based mounting media, that xylene or toluene will evaporate over time, and your media is going to get thicker and thicker, and it kind of creates this sort of silly string effect. So you get these small little stringy guys as you're trying to use your media. So you can thin that down with xylene to prevent that stringing from happening. And when you are cover slipping by hand and you're chasing your bubbles, don't go right over the top of your tissue, because then you're going to scratch that surface of the cover slip, and microscopically you'll see this nice big scrape down the middle of the section. And you can avoid that scratching effect by using soft utensils to chase bubbles, as opposed to like a metal pair of forceps. Special staining and specifically silvers. I recommend that everybody acid wash their glassware with their silvers. In order to acid wash your glassware, it's a 3% acetic acid bath for 10 minutes, and then you follow it with a distilled water wash and let it air dry. You don't have to make a huge batch of 3% acetic acid. In fact, you can make like 50 mils of it and fill a Copeland jar, let it sit for 10 minutes, and then pour that acetic acid into another Copeland jar. And you can actually see that acetic acid start to pull contaminants from the inside of the Copeland jar, and it'll start to discolor, depending upon what type of special stains have been in that Copeland jar before. And until you see it start to discolor, you can use that acetic acid over and over a couple of times. After I am done acid washing, I will cover whatever glassware it is. It can be Copeland jars, it can be flasks, Erlenmeyer flasks, whatever it is that you're acid washing. When I'm done, I will cover it with tinfoil or with parafilm. This does a couple of things for me. It will prevent all those dust particles, again, from getting in the glassware that you just acid washed to remove that stuff. And two, it alerts the person behind you that, hey, this particular glassware has been acid washed. In my lab, I have two sets of glassware, a cabinet that has those that have been acid washed, and then a cabinet for those that have not been acid washed. Now, when we talk about silver stains, I have found over the years that if you slowly heat your silver for impregnation, you will be less likely to flash your silver. And it's really easy to stick your silver solution in the microwave and push the button, walk away, and then you come back and it's flashed. And flashing is that mirroring effect that you get that's attached to your Copeland jar, but then doesn't attach to your slide, or is attached to your Copeland jar and your whole slide looks like a mirror. To prevent that flashing, if it's happening to you continuously, then just slow down your heating process to prevent that. And I found over the years that if you do flash your glassware, you can remove that flashing by soaking your glassware in a 10% bleach and then following it up with an acid wash. So here we have two pictures of silvers. On the left-hand side is a over-impregnated section, and this is what your section will look like microscopically if flashing has occurred while you're heating your silver impregnation solution. On the right-hand side, we have this particulate right through here, that little small black guys in there that are from your ammoniacal silver not being mixed correctly. Remember that when you mix that ammoniacal silver solution, you need to leave a little bit of turbidity in it in order for this to not occur. If you have too much turbidity or you allow your ammoniacal silver to go completely clear, then you may see these small little silver precipitates. Almost all special stains, the precipitate that occurs, it can almost always be prevented by filtering the solutions. You also need to make sure that all of your chemicals that you're mixing are dissolved appropriately. Oftentimes, when we mix our chemicals, we don't allow them to actually go into solution long enough, and you'll get this heavy precipitate that sits on the bottom of the bottle, and that's the stuff that we don't want to get onto our section. That precipitate is going to make your stains blotchy. Of course, we don't ever want to stick a pipette into a bottle and pull out a solution and then we have leftover in the pipette. We don't want to put it back in the bottle unless you're using sterile pipettes. With new techs and new students, I often run into, well, I'll need, 20 mils or 10 mils to flood my slide, but I pulled, 40 mils. Can I put it back? The answer is no. Don't put it back. Always keep in mind that the more a solution is moved from one container to the next container, the more likely it will become contaminated. This is an example of an Alcium Blue 2.5 that was unfiltered or undissolved, and you can see these little precipitation, blue precipitations on the section, and then we could have prevented these by filtering our solution or by making sure that our Alcium Blue 2.5 was totally dissolved prior to use. This is an example of a silver stain that was done in a Copeland jar that had residual Prussian blue staining in the Copeland jar. So we ran a silver, and you know, those Copeland jars that we have hanging around that you look inside and they're blue or they're yellow, and we get lazy and use those. This is the effect that you're going to see. All of this blue precipitate out here is the contaminant that came from the Copeland jar that was previously used for an iron stain and then inappropriately cleaned for the silver stain. Tap water. Our tap water can contain minerals and microorganisms. I test my tap water periodically in order to see what I'm up against. If you're using tap water instead of distilled water for some of your special stains, you can come across cross-contamination. The tap water can contain gram-positive, gram-negative, AFB, and fungus, and iron even. So I will just take a few slides, and I'll dip them in my tap water, and then I'll run a gram-positive, gram-negative, a brown and hops, a brown and bran. I'll run an AFB stain. I'll run, say, a PAS stain, and I'll run a Prussian blue, just so that I can see what's in my tap water. This is an example of tap water contamination. This is actually straight out of my lab here, and it's my tap water test with my brown and hops, and my tap water actually contained gram-positive and gram-negative bacteria. It's not that the bugs are still alive and they get you, but rather they're dead through the water treatment plant, but they're actually still there, and they can lead to false positive results. Recycled solutions. If you're recycling your solutions, your alcohol specifically, I have the gravity recyclers, and they have a filter pack inside of them. If you have any amount of xylene that has gone into your alcohol recycler, then that filter pack starts to break down. So I test all of my recycled solutions. I'll dip a slide into the alcohol that's coming out, let it air dry, and then I take a look at it microscopically. And every bottle that I take out of my recycler, I label it with the test date and the initials of the tech that actually tested it. And of course, we have our record-keeping logs of the recycled solutions that give the date, the solution, the amount recycled, the date it was tested, and the initials of that tech. Here we have a picture of contaminated recycled alcohol. I have two recyclers. I have a clean alcohol recycler, and then I have an old recycler because I have students that at one point in time, one of the students had put xylene into my alcohol recycler. So my filter pack then started to break down into my alcohol. So what you're seeing here is this black precipitate that's all over here is that filter pack that's breaking down and remained in the alcohol. And then this guy that you're seeing here, this pink hued structure, is residual eosin that's remaining in the alcohol because my filter pack had started breaking down. It was unable to remove this from the alcohol solution. So it remained in the alcohol solution, and all of this contaminant showed up on every single one of my slides until I was able to figure out where this was coming from. And as it turns out, it was that filter pack in my alcohol recycler. So that's all I have. I mean, we could go for hours and hours talking about hundreds of different ways to avoid cross-contamination, but there's a few tidbits that I've learned over the years, and I hope that they have helped you. Are there any questions?